I did not get anything.
It's
Alva.
hold on. I'm tagging you.
Okay. Now we got it. Perfect.
Okay.
Thank you.
Yep.
Okay. Fantastic. Welcome, everybody. We're going to be waiting a couple minutes, let people come in. Wait till things sort of even out a little bit, plateau, as it were. But we're thrilled you're here. Get this rearranged a little bit. All right. Great. I know I spoke to some people about this webinar at the American Epilepsy meeting last couple of weeks ago. So hopefully we'll get some draw from that. So we're just waiting for a couple minutes till we see the attendee number plateau just a little bit, and then we will get going. We have a very exciting webinar today. Some really cool information. Hopefully everybody's healthy and ready to get a little bit of time off. Take a little bit of a break. Sorts. Great. Good. All right, well, why don't we go ahead and get started? We've already got a good number of attendees, and this first couple of minutes is going to be introduction anyway. So I wanted to start by welcoming everybody to today's webinar in the series Challenges in Pediatric Neuroscience Research. My name is Adam Hartman, and I'm a program director in the Division of Clinical Research at the National Institute of Neurological Disorders and Stroke, which is one of the institutes at the U.S. National Institutes of Health. I'm also a practicing child neurologist and epileptologist. And so we created this series to help investigators overcome some of the challenges they might face in pediatric neuroscience in areas including neurology, neurosurgery, psychiatry, psychology, neurodevelopmental disabilities, physiatry, critical care, emergency medicine, pathology, radiology, and pharmacology, just to name a few, and there are many others. The ultimate goal is to improve quality of care for our patients, but the more proximal goal is to enhance research that will facilitate improvements in practice. Our webinars are targeted to trainees, senior investigators, and even to those who aren't currently involved in research, making the pie bigger, so to speak. So today we're hosting Doctors Karen David, Corey Kelly, and Chris Boshoff, who are all program staff at NINDS. They're going to be discussing some relatively new and very innovative funding opportunities at NINDS. So first, a few technical notes about the webinar. The session is being recorded and it will be transcribed and archived on the NINDS website, hopefully sooner rather than later. Details about where to find the archive version will be posted on social media and professional society listservs and newsletters. There will be question and answer at the end. To ask one of our presenters, you can click on the, I believe we have the Q&A icon at the bottom of your toolbar. I'll be moderating the question period. I wanted to especially thank Dr. Alva Rosinos from NINDS for all of her technical support in coordinating the webinar, and also the NINDS Office of the Scientific Liaison for all of its support and publicity. So let's go ahead and get started. Our first guests are Dr. Karen David and Mr. Corey Kelly. Dr. David currently oversees the Circuits and Integrated Approaches portfolio of the BRAIN Initiative. She also leads the NINDS Interdisciplinary Team Science RM1 program, which is what she's going to be discussing, and manages the NINDS assigned BRAIN Initiative K99R00 awards. She joined NINDS in 2013 and has served on BRAIN Initiative in various capacities over the years, including as a project officer, as a scientific review officer, program analyst, and currently as a program director. She's also worked on various special projects, including coordinating review activities of the biospecimen Repository Acquisition Committee, or BRAC, for the Parkinson's Disease Biomarkers Program. Before joining extramural research, she did her postdoctoral fellowship in molecular biology and genetics at the Johns Hopkins School of Medicine. She earned her bachelor's degree from the University of the Philippines and a PhD in cellular and molecular medicine from the Johns Hopkins University School of Medicine. Mr. Kelly works in the research operations branch within the NINDS Division of Extramural Activities. He serves as the primary contact for the NINDS collaborative opportunities for multidisciplinary, bold, and innovative research, innovative neuroscience, and partners with Dr. David to implement the program. He brings over 2 decades of public service, business leadership, and research operations expertise. Prior to joining Research Operations Branch, he served as a health program specialist in the NINDS Systems and Cognitive Neuroscience Cluster. Prior to joining NINDS, he served as the Associate Director of Operations for the Stroke and Applied Neuroscience Center and as the Senior Research Scientist and Engineer in the Department of Neurological Surgery at the University of Washington, where he managed a large-scale multidisciplinary research program at the intersection of neurological surgery, bioengineering, and data science to develop and commercialize minimally invasive technologies for the OR and clinical environment to obtain physiological and genetic data to detect, diagnose, and treat neurological diseases and disorders. He is a veteran and served in the U.S. Army as an infantryman. He's completing his PhD in translational health sciences at George Washington University School of Medicine and Health Sciences, where his research focuses on business leadership training for biomedical scientists. He received his master's in medical physiology from Case Western University, Case Western Reserve University School of Medicine, and his bachelor's degree in cellular and molecular biology from Seattle University. So with that, Karen and Corey, please take it away.
Thank you so much, Adam, Alva, and team, and to our audience for this opportunity to talk about funding opportunities for team science. And we'll also be featuring our new team science program at NINDS called the Combine Program. So this will be a team presentation between me and Corey Kelly. We are co-leads for the Combine Program. So I'd like to start with very brief introduction from us in the context of our roles in the Combine Program. Go ahead, Corey.
Thank you, Karen, and thank you, Adam and Alva. And most of my introduction was covered by Adam. I'll just add that Karen and I work together to implement this program, and we hope to hear your questions today. Thank you.
Thanks, Corey. And I'm a program director for the BRAIN Initiative and NINDS. And this combined program came about through an NINDS working group that studied the strengths and weaknesses of different structures and models of team science funding across NIH in various programs from different agencies. So with the combined program, we support projects that are multidisciplinary, bold, and innovative. So for this talk, we'll highlight the opportunities that collaborations bring us. We'll also talk about what are your options for getting your teams supported at NINDS. And then Corey will be talking about the combined program and close it off with next steps and Q&A. And also, as Adam mentioned, feel free to put in the Q&A text box your questions as well. So this talk is about funding opportunities for projects that fall within the NINDS mission, and we will not be discussing in this talk what those topics are, also because of the complexity posed by some scientific topics being of shared interest across the different NIH institutes. For example, NICHD has a shared interest with NINDS on certain topics in the pediatric space. And here is where we as program officers can help. We can help assess whether your topic is a good fit for NINDS or for specific funding opportunities. So please reach out to your program officer and we can help you, direct you to the right institute or funding opportunity. And also it's helpful if you share a project summary description or a draft of aims. Now, before we talk about how NINDS supports team science, I'll start with a quote from our director, Dr. Koroshetz. We are stronger together through interdisciplinary team science. And team science is particularly important to neuroscience. Given the complexity of the nervous system and the challenging questions about health and disease, no one person or lab is going to have all the expertise and tools needed to pursue challenging goals. Team science provides us the opportunity to pursue really difficult goals in a more comprehensive and rigorous manner, and also provides us the opportunity to take advantage of our increasing knowledge base and armamentarium of technological capabilities to neuroscience. Okay, so there are several ways that team science is supported at NINDS, and I'll be going through an overview of the differences across these NINDS team science funding opportunities. Okay, so to start, the blue circle here is meant to symbolize a single project or component. And so for the PO1 program project, we depict its multi-component structure where you have a certain number of projects plus cores, and these components would have interrelated and parallel goals. Now, a multi-PI collaboration can also come in through a parent R01. And in contrast to the PO1, this will be a single project with one set of aims. Now, in contrast to this, we have the RM1 combined team science structure, which emphasizes integration. Basically, how disciplines are merged towards the pursuit of one single challenging goal that can only be done through team science. So no multiple aims here. Now, the PO1 and RM1 are team-specific and team-centric, allowing for inclusion of elements and resources that are team science specific. And the PO1 and RM1 are also reviewed in its own study section, so team science projects are reviewed alongside like projects. This is in contrast to the R01, which is reviewed in multiple study sections, and applications range in different sizes and scope. As for the scale of the RM1, in general, it would be larger than the parent R01 and similar to or larger than the PO1. Now to recap those points, but in the perspective of the applicants, how do you decide which funding opportunity is the best fit for your project? Just a reminder also that this information is specific to NINDS team-based applications, as other NIH institutes will have a different set of funding opportunities. So first is to look at what is the best structure and scale for your science. So some studies will work with multiple aims, or by just having one aim or goal, or being a set of projects and cores. Now, some general considerations about budget. Just know that for R01s greater than 500K direct cost, and for the PO1s, you would need prior approval from NINDS before you apply. And for the RM1, you can reach out to me and Corey to discuss your project. Now, when are you thinking about applying? So the R01 and PO1 have 3 receipt dates per year, whereas the combined RM1 has 1 receipt date per year. And another thing to pay attention to is the responsiveness and non-responsiveness criteria described in the funding opportunity, as this vary across these different funding mechanisms. So for example, the PO1 and RM1 do not accept specific types of clinical trials, and RM1 has restrictions on certain types of studies that Corey will be further describing. Now, speaking of specifics about the combined program, because the combined program is new to NINDS, we'd like to take our time to talk more about its features. So I'm passing it on to Corey. Go ahead, Corey.
Thank you, Karen. So as Karen mentioned, now that you know kind of the differences between our combined RM1 program, the PO1 and the multi-PI R01, we'd like to talk to you a bit more about our program. This is a pilot program that we started a couple of years ago. We changed the NOFO last year, so we just wanted to highlight a couple of changes between the original announcement and the current announcement. Those are highlighted in yellow. I'll go through each of these topics in a bit more detail, but here on this first slide, just at a high level, what is this program about? The Combine program is looking for transformative goals that can be achieved in 5 years that are fully within the NINDS mission. We're looking for bold, impactful, and challenging ideas that will change the paradigms of how we understand and approach neuroscience, and they must be achieved only through interdisciplinary team science. As noted, a change for this year in the current NOFO is that multiple aims are non-responsive, and we'll talk about this a bit. As well, the Combine program requires 3 to 6 multiple principal investigators that each bring distinct viewpoints or expertise to the program. And it's generally anticipated that each PI will provide at least 2.4 calendar months per year. And PIs on the Combine program are subject to the NINDS SCR policy, Special Counsel Review Policy. Discussing the scope and complexity, Karen discussed kind of the differences between these different mechanisms and what sets them apart as far as their approach. As far as the budget for these Combine applications, we anticipate seeing applications between 500,000 and 1.5 million direct costs per year. And last, as Karen did mention, integration is a critical element of the Combine program. So we expect to not only see integration between the investigative team and the other personnel, but also see that the team science approaches integrated with the research strategy is demonstrated through the team management plan and the plan for enhancing diverse perspectives. So speaking towards the goal for the Combine program, as I mentioned, these should be transformative goals that can be achieved within the 5-year scope of the RM1 program. These goals should challenge existing paradigms, overcome longstanding roadblocks to progress, and to develop new synergies between our different scientific fields. And these should be challenging so that they can only be achieved through a team science approach. To justify your application for the RM1, we really want to understand why team science approach is needed, and that this couldn't just be achieved through individual or parallel or a series of efforts. This is considered a high-risk, high-reward program. So we do, again, with the innovative aspect, expect to see some degree of risk compared to maybe a more traditional R01. But feasibility will be assessed by the study section reviewers. So you should mitigate the risk in the research strategy through either preliminary data, the strengths of your team, or other options. The approach to the Combine program, as I mentioned, it's 3 to 6 multiple PIs. Everyone should be working towards a singular, focused, transformative goal, and each of these PIs should bring a distinct field or expertise. We don't want to see overlapping expertises. Every single person that's on this application should be essential and distinct. And this should be reflected in the aims, the budget, the leadership plan, and the team management plan. And we've presented a graph to the left to kind of talk about what we're talking about when we see integrated efforts as well as equitable efforts. Unlike a multi-PI or R01, there should be even efforts between all the PIs in terms of their contributions of science and their bandwidth. We don't want to see a collection of individual efforts where PIs might be working in parallel on kind of siloed projects, or a series of projects that could maybe otherwise be funded as a series of R01s. So we really want to have distinct expertise that comes together integrated upon this single focused goal. And to that point, we kind of use a thought exercise that we call the deletion test. And as you can see, we try to describe this in this house on the left. If you were to remove any single piece of this puzzle, or the investigative team, then you could not achieve this transformative goal. As we mentioned, one of the distinguishing factors between the P01 and the multi-PI R01 is that multiple aims are not responsive. We did this for a couple of reasons, but primarily with the complexity of team science and transformative science, often approaches don't fit in this kind of traditional series of interdependent aims. We, in fact, want your work to be integrated, therefore we want it to be dependent. And so thinking towards this integration, you'll see we've highlighted it 3 times on the slide because integration is such a critical element here. If it's a series of individual efforts, we could fund it as an R01, or if it's a series of interrelated but parallel projects, it could be funded as a P01. So we really need to justify why all these investigators need to come together to work in the same processes to achieve the same single goal. And we kind of describe the activities that bring the investigators together as glues. These are the pieces or the knowledge or the personnel that bridge the different disciplines that will help you overcome the transformative gaps and achieve your focus goal over the course of 5 years. And speaking towards the scale, again, comparing to existing programs. Again, we want shared leadership. We want equitable leadership. We want distinct expertise between the multiple PI leadership team. Because of the additional complexities and sometimes resource requirements of team science, the scale of this program will likely benefit from including a program manager or a program coordinator or a data scientist or whatever additional personnel you might need to tie your team together and facilitate your progress through the grant period. And this can be requested in your team management plan and reflected in your budgets. There are some changes to the application to reflect the additional scale of these proposals. So, and I'll get into these a little bit more on the next page, but there is an increased 15-page research strategy, as well as 3 additional attachments. So again, the hallmarks of the Combine program: transformative, innovative, and integrated. The key component is to address how will the project challenge existing paradigms, overcome longstanding roadblocks to success, or develop new strategies between scientific fields. And we want you to be intentional about how you're integrating efforts across activities, across teams, and across disciplines so you can share knowledge and achieve your focused goal. And lastly, we do expect some risk in this innovative, high-risk, high-reward program, but you will need to address the feasibility in your research strategy. So again, going to these application differences, we have a 15-page research strategy. Again, multiple aims are not appropriate. And we have a simplified structure that we will request for the research strategy, which is simply the importance of the research and appropriateness for the NINDS Combine program, and the approach and experimental design. I will note, feel free to put whatever subheaders under these, but these are the high-level headers. As I mentioned, we have 3 other attachments specific to address the needs of team science. The first is well-managed team interactions, and this is addressed through the team management plan, which is distinct from the multi-PI leadership plan. The multi-PI leadership plan specifically discusses the governance between the PIs, what happens if someone leaves, et cetera, whereas the team management plan discusses the interactions between all the teams and all the different members that are contributing to the project. The second is timelines and benchmarks for success. Something like a Gantt chart is very typical here. And then last is evidence of commitment to diversity, equity, and inclusion. This is a newer attachment to NINDS and to the NIH, the plan for enhancing diverse perspectives. We have had this on our program the entire time. We define diverse perspectives very broadly at NINDS. We have a lot of information on our website about this. But the key thing here is we don't want you to just list activities that will contribute to enhancing diverse perspectives, but they should be intentionally integrated with your research strategy to further the efforts of your science. A lot on this slide. I'm not going to read it all, but just a critical point here, as Karen mentioned at the beginning of the presentation. One thing that's distinct about our program compared to maybe an R01, where we can share interests and co-fund things that might be of interest to other institutes at the NIH. Applications of the Combine program have to be entirely within the NINDS mission. Again, we're not going to get into the details here today, but we're happy to discuss these with you if you inquire. At a high level, the other non-responsiveness criteria to our program is programs that are a better fit for other institutes or specific funding opportunities. As we mentioned, this is a pilot program with fixed budget limits. As such, if we have funding opportunities available that would better fit your program, we would refer you to them. So you can send your application to the place where it would be most likely to be funded. And then a couple other things: applications of which the primary goal is to create, maintain, expand, or disseminate resources or infrastructure. These types of platform building or resource building approaches, we have better opportunities that you can apply to at NINDS. And then lastly, very nuanced, but considerations about translational and clinical types of applications. We are interested at a high level in translational and clinical research, but we do have a lot of specific NOFOs for these types type of applications. So what we are interested in accepting into the combined program in this space is mechanistic clinical trials or basic experimental studies involving humans, BESH clinical trials, but they must have a transformative single focused goal across the 5 years, as well as all the other requirements of the combined program. So the last thing on the slide you'll see highlighted in yellow, non-responsive applications will be withdrawn prior to review. So we're very eager to provide consultation to any interested applicants and provide you some feedback to make sure that your application, if submitted, will go to review and potentially be funded. So I'll hand it off back to Karen to discuss some of our programs that were funded in the first cohort.
Thank you, Corey. See, I'm not muted, right? We're excited to share that we awarded our first set of projects, and the diverse topics covered here include basic, translational research, and various disease topics, with all integrating different disciplines. So I'm highlighting 2 projects here. One is the project on the glucose transporter deficiency syndrome, which is most often associated with medication refractory epilepsy and movement dysfunction. So that's the 4th project listed here. And in this project, we have 5 labs who are combining their expertise to look at the metabolic and neurophysiological aspects of this disease in both mouse models and human studies. Next slide, please. And the second one that we'd like to highlight is this project that just released a really nice summary of what the award is about. And here's the link to that article. And in this project, we have 6 teams bringing together scientists in pediatrics, genetics, bioinformatics, and psychiatry to understand how and why cells die in 2 lysosomal storage disorders that affect the brain. So the central hypothesis of this project is that the disease pathogenesis is due to the changes in the communication between the lysosome and the rest of the cell, rather than the storage-related issues itself, which is the current prevailing view. And so I'm passing it over to Corey to wrap it up.
You can. Okay, we'll take some questions today, but if you have additional questions, please do view the NOFO, which we've listed on the slide, as well as visit our website, which has a bunch more information beyond the NOFO, as well as a list of frequently asked questions across various subjects, which you can see on the right. And lastly, if you have any other questions, you can feel free to reach directly out to Karen and I at ninsteamscience@nih.gov. Thank you.
Thanks a million, Karen and Corey. I think so. What we'll do, because the 2 topics are a little bit separate, is spend about 5 minutes answering questions, and then we'll do the same thing for Chris's presentation. And then if there are joint questions afterwards, we can do those. So the first 2 questions are they're sort of similar. So the first question is one goal versus specific aims. I think there might be a little, we're all used to writing specific aims and reading specific aims. So the first question is one goal versus specific aims. And then the second question is, can you explain a bit more the bullet point on one of your slides about aims being non-responsive? Who wants to, you can answer that sort of as one, I think.
Yes, I can start, and then Corey can chime in if I miss anything. So our experience in the past year has been that the team struggled with proposing cohesive and integrated projects because of the usual view that multiple aims cannot be interdependent. And so for this new version of the combined program, we emphasize pursuit of this one focused goal or aim, which we use interchangeably, and consider multiple aims to be non-responsive. So what we would just mention here that how does this look like practically in your application? So we understand that the pursuit of your scientific goal would likely entail structuring your project into pieces. And you're free to call these pieces how you see fit, just not as multiple specific aims because of what's attached to what we call an aim, right? So for example, you can call your pieces approaches or even sub-aims, as long as these pieces are essential to the goal and are tightly integrated.
Great, thank you. Next question, this might be an easy one for you to answer. When will be the next MPI submission?
For the Combine
I think,
program,
yeah, I think it's for the combine
the
program. yeah.
next submission date is October 2024.
Great, thank you. Okay, the next question is, what type of endpoint are you envisioning? Curing a disease, first in human studies? What do you see as the endpoint of this, of an individual grant application?
So this is similar to an investigator-initiated application, meaning that the investigator or team of investigators specify what is that transformative goal. So this is something that we leave it to you to articulate in your application, and peer review will then evaluate whether what you're proposing is transformative or pushing boundaries, as we define in how we define transformative in this program.
Fantastic. The next question, I'm not quite sure I understand it. Wondering if you could speak to the differences between the NINDS Interdisciplinary Team Science Grant RM1 from RFA NS 22036 in comparison to the Combine RM1 under the same.
Yeah, no problem. The numbering didn't make it easy. The Combine is actually NS 23027. But these are the same programs. So when we initially made the program, it was the 036 Interdisciplinary Team Science. But moving forward is now the Combine program, which the most recent FOA is NS 23027. But the same program, new name.
Yes, use the most recent one.
Please
most
do the
recent
one
one
that's on
because
the
of the
slide.
aims.
Right.
Yeah.
Okay, two quickies, and then we're going to move on to Chris. Do the labs or teams in the combined program have to be from the same institution?
We have no preference whether the investigators are at the same institution or different institutions. We just, again, want to reiterate that the efforts between the PI should be distinct and completely integrated as necessary to achieve the single focus transformative goal.
Right, and I'll just add to that is what we care about is what do you need for the science, right? What is that expertise? And then what we care about is that the expertise you need is represented in what you're proposing.
Great. All right, last question, then we need to move on to Chris. How much, if any, preliminary data is expected?
Right. I can start. So I would say the way we tell reviewers is feasibility. You can look at feasibility in a holistic manner, meaning it could be a combination of things of which preliminary data is just one of them. It could be your track record, your feasibility, your preliminary data, how you're managing the risks in your approaches, in your premise. So I would say it's one of the components of feasibility, and we look at feasibility. So that is a review criteria, and basically, just to reiterate, preliminary data is just one of the many pieces that you can show for feasibility evidence.
Perfect. All right, stick around, gang. We now have our second speaker, Dr. Chris Boshoff, who is a program director in the Division of Translational Research at NINDS. Dr. Boshoff joined NINDS in April 2014 and currently serves as the scientific project manager for the collaborative research to enable and advance translational enterprises for biologics, or CREATE Bio. He also facilitates translational efforts of intramural innovations. Prior to joining NINDS, Dr. Boshoff worked at the United States Diagnostic Standards, where he was director of program management, overseeing regulatory and reimbursement strategies for clinical diagnostic development and early phase oncology clinical trials. Prior to that, he worked at Navi Biopharmaceuticals, and it was developing a smoking cessation vaccine, and at the Foundation for the NIH, where he was responsible for the immunology and inflammation biomarker efforts. He also worked at Technical Resources International, which is a clinical research organization focused on oncology drug development. He gained his core experience on biologics and diagnostic devices development at Abbott Laboratories and its subsidiaries, where he spent 8 years after receiving his PhD in virology from the University of Natal in Durban, South Africa. Welcome, Chris.
Thank you for that introduction, Adam. I just want to make sure, can you all hear me well?
We're good.
Thank you.
You might want to flip your slides in a presentation mode, though.
Let me see. Is that there? No, I don't see it.
No, we're not.
Is that better?
Nope, it's the same.
Well, I can see it. Better?
Try
No,
sharing a different screen. You're probably sharing
Yeah.
the wrong screen.
So in the meantime, while Chris is getting that straightened out, if you have questions for Chris or if you have them for Karen and Corey, please use the Q&A tab down at the bottom toolbar. And the slides will be available. We're going to post the slides, PDFs of the slides, onto the website, the webinar website. Everybody who registered for the conference will get that link. And then, but it's also publicly available. We'll have both the videos that have transcripts as well, or that have the text, and the slides themselves. And if you have obviously additional questions, you have contact information for Karen and Corey, and you will have it for Chris as well. Chris, you want to? Yeah, you want to just why don't you just go ahead and present, because I don't want to
Can you see
take
my
up too much
slide?
time. We can see your slides. It's just not in presentation mode, but that's okay. Why don't you just go ahead and do your presentation?
I'm
as
sorry
is.
about this. I'm very
That's
grateful
okay.
to present the NINDS Urgent program. Urgent is an acronym standing for ultra rare gene-based therapies. It was selected with a little bit of deliberation behind it. We wanted to design a program that is responsive to the needs of the patients we hope this program will serve. It's a relatively new program. We launched it exactly 2 years ago in December 2021. And it didn't happen in isolation. We were very much aware of the context or the background in which this occurred. And as you know, biotherapeutics make up an estimated 25 to 30% of the therapeutic agents on the market currently, and it is indeed expected to increase. At that time, we saw some very encouraging FDA approvals, such as Spinraza for the first ASO treatment for SMA, that got approval in 2016. After that, Luxturna, the first AAV gene therapy treatment for a rare retinal eye disease, approved in 2017, and then Zolgensma, another gene therapy for SMA, approved in 2019. And now, of course, we've seen some other exciting developments in this field since then. And then, so of course, future expectations in terms of new designs and platforms are expected to poised to accelerate the research and clinical development and facilitate further approvals.
Hey, Chris, I'm going to share my screen because your slides aren't advancing.
Oh,
I happen to have your slides, so I'm going to go ahead and share my screen for you. Let's see if that works. Put this into slideshow mode. Okay. Let's see if I can get this right here. Okay. I do these one at a time. Okay. Can you see the slides, the background slides?
No, Adam, you're sharing your screen.
My other screen?
Yep.
Okay. How's that?
Not
It's
yet.
still the same. Oh, now you got it.
That's
Perfect.
good.
Okay.
Yeah, thank you, Adam. So yeah, this is the background slide. Next slide. And then, of course, at the same time, there was this inspiring story now known as Mila's story, a little girl who was diagnosed with a rare form of Batten's disease. Long story short, she got referred to Boston Children's Hospital. They identified the genetic variant. They did some planning activities looking at the variant and establishing patient cell lines with the idea to develop an ASO for this patient, that's N=1 type of scenario. They went through the identification of different heats, validation of the ASO functionality, and then eventually started the first in human clinical trial for this patient. And this was really inspiring for us as well, and it really forced us to think through what can we do here at NINDS to support this kind of approach even more. And so next slide. We were very much aware of the challenges to do that for it, and I'm just highlighting 2 or 3 here, and that is the lack of access to manufacturing capacity, especially when it comes to gene therapy, and then therapy development expertise and regulatory guidance may not always be readily available to these investigators. Lack of available infrastructure for academics and small businesses to do this kind of thing, especially these later stage translational projects involving manufacturing, toxicology, and clinical testing. And then there's also the challenge of these ultra-rare disease patient population. There is no commercial incentive to develop these therapies. And so we had to think through how to make this available to these patients. And then, next slide. Yes, next slide. So we asked the question, why now? And I think we came up with very obvious answers. Many of these diseases are within the mission of NINDS currently, and they are monogenetic and amenable to personalized therapy approaches. Recent scientific advances suggest that the great promise of these modalities, especially for ASOs, and there is no, at that time, there was no current NIH program available to support this type of therapy development. And the majority of investigators lack the multidisciplinary support to advance these ideas from the bench to the bedside. And then there was also a great deal of developments with regards to the creating a favorable regulatory environment to make these things possible. Next slide. So the strategy that we came up with was to establish a mission. The mission of the network is to provide resources and funding, not just funding, but also the resources for investigators to develop these gene-targeted therapies from bench to bedside. And the plan was to build an infrastructure that includes access to these contract resources for various activities, including manufacturing, pharmacology, large animal studies, an external oversight committee staffed by individuals from industry, especially people with the experience of drug development, a centralized data management system to harmonize the data that we will create, and then make available consultants in different areas, such as toxicology, pharmacology, regulatory, IND-enabling studies, etc. And then to facilitate the formation of a network, not just an individual program, but a network of clinical sites to execute these first-in-human clinical trials. Next slide. So the goals of the urgent program are to accelerate the advancement of these discoveries from idea to discovery to the clinic, provide the resources and expertise not readily available to these applicants, deliver therapeutics to patients with ultra-rare neurological diseases, and standardize and harmonize best practices. So the urgent program will support these principal investigators with a lead gene therapy from start to finish within 3 years through planning, manufacturing, IND-enabling studies, and ultimately an IND submission to the FDA so that they can initiate first-in-human clinical trials. Next slide. So the scope of the program, it is focused on ultra-rare neurological diseases, and the way we define it for this particular program is fewer than 6,000 patients in the US, and that equates approximately to 1 in 50,000 people. And the modalities that we regard as within scope or meeting the requirements of a gene-based or transcript-directed therapy would be modalities such as oligonucleotides, like ASOs or small inhibitory RNAs, viral-based vector approaches, genome editing, or any other gene-based therapeutic. It may even be a small molecule such as a splice modulator, as long as it is gene-based or transcript-directed. Next slide. So here is the program that we came up with 2 years ago. It was designed to support the development of these gene-based and transcript-directed therapies for ultra-rare neurological and neuromuscular diseases. It was designed for a phase funding mechanism with multiple entry points. The first entry point is the investigator just want to plan or do a gap analysis in terms of what they have and where they should go next. It could, the second entry point, when they already have a clinical candidate and they are ready to embark on therapeutic development. And the third entry point would be after filing an IND and the IND clears and they can do a first-in-human clinical trial. And then we will support in-kind access to R&D contract and expert consultant resources to facilitate this. And it's a late-stage program, it's an accelerated program, so the idea is that the investigator should reach the IND within 2 to a maximum of 3 years. Next slide. So the way it was structured is we launched a program with 2 grant mechanisms. The one was an EXO-1, PAR 22028. This is a mechanism allowing an investigator only access to the contract resources, and then they can do planning activities, strategic planning activities, and therapy development. The second grant mechanism is a UO1 mechanism, PAR 22030. It's a cooperative agreement, and for this mechanism, they have 2 to 3 years to get to an IND. And then very recently, we added on a clinical network embedded within the NeuroNext program with a centralized data management system available. And once the IND clears, we can fund the first-in-human clinical trial through another mechanism called OT. It's not a grant, it's not a contract, it's another mechanism standing for other transaction. It affords us the flexibility to quickly move towards clinical testing after the IND clear. Next slide. So this is a closer look at the clinical trial network within the NeuroNext system. We have clinical trial sites supported by a clinical coordinating center and a data coordinating center. There is a dedicated DSMB. We make consultant or subject matter experts available to the investigators in any aspect of the program, and all of this together then forms what we call the NINDS Gene Therapy Consortium, and it is overseen by the external consulting board with experts in various disciplines such as clinical trials, manufacturing, regulatory, etc. Next slide. So just a quick word on the contract resources that we have. We launched a program with these contract resources. We have prime contractors, Leidos, ABL, and Battelle. They have the ability to subcontract specific areas in case they cannot do the work, such as manufacturing or toxicology. A successful applicant then has access to these contract resources. We form multidisciplinary teams around the PI, and we build and manage a project team. We coordinate and the contractors, and we provide technical direction and guidance. And so with these prime contractors and the subcontractors, we move from assay development, toxicology work, upscale of the manufacturing, and the first-in-human clinical trial within the network I just described previously. Next slide. A quick word on the consultant network that we also have. At the moment, we have approximately 25 consultants in various disciplines available to successful applicants. And currently, we are recruiting experts in oligonucleotides, AAV gene therapy, and assay development. And these people have expertise in various disciplines such as CMC, toxicology, regulatory affairs, medical writing, etc. Next slide. Just a quick word again on one of the funding opportunity announcement, PAR 22030. This is the UO1 mechanism. In terms of the entry requirements, it is a very late-stage translational program. We want to see compelling proof of concept data showing the feasibility and the rationale for the candidate selected. And the program director or the PI should have identified the gene-based transcript directed therapeutic clinical candidate. In other words, all the optimization work should have been done already. There should be a fully optimized clinical candidate, and they should just be ready to do the IND-enabling studies and the manufacturing. And the PI, we prefer that the PI already had some interactions with the FDA, especially in terms of the design of the toxicology studies, because those studies are notoriously expensive. And the PI should be able to provide an outline for the future clinical trial. Next slide. So typically, the type of studies that we would support would be these late-stage and non-clinical development activities, including but not limited to things such as manufacturing, qualification or validation of any assays needed for the IND-enabling tox studies, additional efficacy studies, assessment of off-target effects if needed, evaluation of the utility of the pharmacodynamic or target engagement biomarkers, completion of all the clinical trial planning activities, and the actual preparation and filing of the IND package. Next slide. It is a cooperative agreement, and that means that NIH program staff have significant involvement and leverage on the actual management of the project. We form what we call the multidisciplinary team around the PI, the contractor, subject matter experts, and we manage the program. We meet on average every other week. It's very intense, and we make sure that the milestones are realistic and feasible, and we manage the program towards IND submissions. Next slide. At the moment, we have almost 4 projects in the pipeline, 2 of them AAV gene therapies, and a small inhibitory RNA project, and we are about to launch another ASO project early next year. And we have 3 receipt dates every year: February, June, and October. So, it's a growing portfolio. Next slide. We do have a website, and I invite you to look at the website. There are lots of good information you can find about the program, resources, etc. So please take a look if you are interested in the program at the website. And then I think the next slide would be my final slide. This is how you are able to contact me if you have any questions about the program. Thank you, Adam.
Great. Thanks, Chris. I'm actually going to stop sharing so I can get myself situated. So thank you to all of our presenters. And so if you have any questions, please post them in the Q&A. If you look along the bottom of the toolbar in your Zoom window, you can see that. If you have questions for Chris, if you have questions for Karen or for Corey, please post them. Thanks to all of you. This is absolutely fantastic information. I can tell from the questions that are being asked that there's a lot of interest. I know I get asked about both of these programs quite a bit. And actually we were at a gathering with a patient advocacy group touring a very interesting facility, and the urgent program came up in one of the conversations. So this is just absolutely fantastic. Thank you.
Adam, do you want to address the question that Corey answered privately, so that just in case someone else has the same
You
question?
can do that. Yeah, sure. So the question was, can you comment on the current and future composition of the review panel, given the interdisciplinary nature of the RM1 funding mechanism? How does this differ from existing study sections? Corey, you respond to that. Do you want to say a word about that? I don't want to necessarily read your words.
Yeah. Thanks, Adam. Thank you for the question. Yes, for the Combine program, we convene a special NINDS study section, a little different than the typical study sections. So each application is reviewed by an increased number of reviewers. We specifically identify the reviewers that are necessary to cover kind of the breadth of expertise that's expected in these Combine applications, as well as additional experts in team neuroscience. And that way we can kind of have a good balance of addressing balancing the specific scientific nuances of the application, balancing those with kind of the greater needs and understanding of team science and its capabilities to achieve those kind of things. And we had absolutely phenomenal set of reviewers for our first cycle, and they really matched the points of emphasis that we provided today with how they review the applications.
I agree. And just to quickly add, for people preparing their applications, the audience here will be a blend of generalists and specialists. So think about that when preparing your application.
Thank you. So we have a few questions, and we'd like to give people, we don't want to run up to exactly the top of the hour. We want to give people a minute or two to run to their next meeting. This has been fantastic. So Chris, this is for you. Most important deadline for January or February in the urgent program proposal process?
The most important deadline. The next receipt date is in February.
Great.
So I recommend that a potential applicant talks to me about the program, and that we can work together to make sure you have everything in place to submit a successful application.
Perfect. And then this is a question for both groups. Are RM1 and urgent geared towards senior investigators, or can early stage investigators or ESIs able to apply? Chris, you want to
So-
take that one first?
Regardless, it doesn't matter.
Okay.
Just meeting
Yes.
the entry requirements of the program. That's the only requirement.
Karen?
Kind of similar to Chris, what we care about is what is the expertise needed, and is that person, whether it's ESI or a senior personnel, is providing the appropriate expertise. And another thing to note is one of our other attachments that we require is the plan for enhancing diverse perspectives, where we welcome diverse perspective that would include, for example, different career stages in your team.
Great.
And I would,
Okay.
sorry, Adam.
Sorry.
Yeah, just one other point about the ESIs. It is important to know, if you are an ESI, you can apply to the Combine program, but if you are funded, you would lose your ESI status.
Great. Next question, have you evaluated to add neuroimaging methods to find new kinds of biomarkers?
I'm
I
not
think
sure
that's
who
for
this
you
is
guys.
targeted to.
Yeah, why don't you take that, Corey?
So I would say at a very high level, anything that would kind of be looking at neuroimaging and biomarkers, we would in most cases refer to the NINDS biomarker program. Particularly, we have an R61, R33 research project grant for exploratory research and biomarkers. So that's probably where we'd refer you in our Combine program. Unless you were, for say example, investigating like fundamental mechanistic understanding that could then lead to future biomarker research. But if you're just seeking to develop biomarkers in and of themselves, we would refer you to those other programs.
Great. Question, is the UX1 still going to be offered?
Maybe
Not familiar with
that's
the
a question for me.
UX1.
There are two funding opportunities for the urgent program currently. There is the EXO1. It is only access to contract resources. There is no funding associated with that. And then there's the UA1, and that's funding plus contracts. And then I may add that there is also now that third entry point where if you already have a cleared IND, you can directly enter through the OT mechanism to enter the Neuronext clinical trial network.
Great.
I
Thank
hope
you.
that answered that question.
I hope so. Yeah, I'm not familiar. I've never heard of a UX1. Can a proposal have preclinical and clinical research be combined?
So for the RM1, we definitely encourage bridging across different disciplines, and that could be, for example, bridging preclinical and clinical. But we can't be definitive about this until we see your specific aims. So make sure to contact us at NINDS Team Science at nih.gov, and Corey and I will help you go through your aims and see if it's a fit for the program. Not aim, one aim.
One aim, right. Okay, last question. This one's for you, Chris. Are there specific CROs or NIH-funded large animal studies groups recommended for the tox studies in urgent?
Recommended, yes. So we have the prime contractors. Some of them can do it themselves. Others prefer to subcontract. But to answer the question, we do have the capacity and the capability to sponsor that kind of studies within Urgent.
Great. All right. So I wanted to thank our presenters. That was absolutely fantastic. The session was recorded and it's going to be transcribed and archived on the NINDS website in the coming weeks. Details about where to find them will be posted on social media, professional society listservs, and newsletters, and we'll be sending it out to everyone who registered for today's webinar. Real huge thanks to all of our presenters and to Alva for helping us navigate some of the aspects here. Thanks a million, and stay tuned. We hope to have more challenges in pediatric neuroscience research webinars in the next calendar year. So stay safe, stay healthy, and happy holidays. Thanks, everyone.